Good afternoon. It is my pleasure to be here. First of all, thanks to the organizers for giving me the opportunity to share with you what Syngenta is doing as a research effort in order to come with future solutions to address the abiotic stress management problem. So in the couple of, or the next 20 minutes, I'll be, sorry, can I use the pointer? Is this, yes, fantastic, and yes. Okay, so in the next 20 minutes, I'm going to be giving you a sort of overview around, first of all, what are the grower needs in terms of abiotic stress management. I think it became already very obvious from different sources that this is a real unmet need for the grower. We do need, as a company in this business, to as well contribute to generate solutions for the future. And I'll be touching upon those specific issues. Then what is our approach within R&D in Syngenta in order to try to tackle those needs? And of course, I'll go into as much detail as I can and give you a sense of what research is occurring within our organization in order to really push forward the different technologies to address those. And of course, it does involve a lot of capable building and expertise in order to be able to progress this complex area. And finally, last but not least, I'll give you a sense of what is our current market products we have, as well as, most importantly, our chemical and genetic research pipeline, okay? So with this, this is a slide which is really an attempt to give you a very high-level view of the size of the yield losses in terms of growers' let's say, loss of income, and you can see that it goes up to $200 billion annual crop losses across the different stresses that you can see here across the different major crops. And without going into much detail, you can see very clearly that the drought stress is an enormous issue and stress for the farmers, and certainly the drought, when it's involved in the corn culture, is really a very big issue. So you can well see that different stresses are impacting in different levels, different crops. And what we are doing with this kind of information really is try to define for Syngenta what are the top, let's say, 6 priority areas in which we should be investing, and how does this inform our research strategy, right? So an example will be a very obvious one, which I'll be taking in the course of the presentation today. It has to do with a drought event during the flowering stage in corn, which has a huge impact on the corn yield at the end of the day. So of course you can always, you can imagine that by somehow combining the different stresses, the different crops and the different crop cycles, you end up with a pool of some big problems that needs to be solved in the future. Without any further, this is just to let you know the way I'm going to proceed. I'm going to show you a couple of things related to our natural products effort, and this will be exemplified with biostimulants, and those are actually mixtures of amino acids. These are already in the market, and I'll give you a sense a bit of the performance of these. Another example has to do with chemistries, and the chemistries I'm talking about small synthetic molecules, and we do have a research project pipeline that I'll be touching upon. Of course, this will impact the market in a long term. Finally, last but not least, we do have an effort in the genetic solution through certainly GM, genetic modified crops, but certainly more into native traits. And we do have already in the market a product that we call the artesian lines, and I'll touch upon those as well. But we are certainly very much interested in the genome editing approach, which shows very promising future in terms of even replacing the GM technology at some point in time. So of course this is also impacting the long-term portfolio. In order to be able to deliver all those different technology in the future, we do need for sure to have expertise and to have capabilities in order to progress on those different aspects of our research. So I'm going to start, as I said, to mention our natural product we do have in the market, which is called the Sabayon. I have to say upfront that this is not a research innovation coming from our labs. This is a product which is a natural source coming from a third-party company that we have extensively characterized for its potential in mitigating abiotic stress. So this is just one example where we do address a specific problem of the rice farmers. It is all about the transplanted shock when you are actually moving your rice seedlings from the nurseries to the field, and they are submitted actually to a cold stress. And this is very much impairing the yield at the end of the day. So to make the story short, Sabayon is able to really increase the shoot dry weight by 13% under our controlled environment. And you can briefly see here how does it look. This is actually pictures which we're taking in my lab, where you can see in Stein we are equipped, and I'll touch upon those capabilities, with quite a substantial phenotyping platform, which allows us in this specific case through image-based phenotyping to quantify the recovery of the plants after stress and treatment with these specific molecule as compared to control. So essentially some numbers here. We certainly have a very good correlation between the shoot dry weight and digital biomass, which allows us then to say that, after the treatment, and you can look into the table on the right corner here, you can see, please focus on the first line, that this is the optimal concentration after the rate dependency study, which shows quite a significant, and this is our best result, showing the advantage to apply Sabayon in order to help the rice seedling plants to recover. So I'm not going to go into details of the experimental design, but certainly these are robust and reproducible effects that obviously a pre-requirement to be able to share those with yourselves and others and to sell the product in the market. Of course, you know, the greenhouse is always a very informative experiment, because it certainly, in one hand, allows us to somehow inform our field trialists on the best rates to be applied for this specific problem. And then you can see, of course, how does this Sabayon molecule performs, or product performs, under field environment. And of course, this is what we're looking for. So definitely we can see an extensive number of field trials were done in this specific case, that you have an increase from 10 to 15% of the yield as compared to the untreated control. So it's already in the market, and it has been extremely successful. Now we're extending this to other crops, and you can see well in the title, it has been extended to vegetable, rice, and specialty crops, for which we have as well demonstrated quite a good performance. So moving into something a bit more exciting, if I can say so, as a researcher, because then this is all about research we are doing internally in our groups. Let me tell you something about the chemistry small molecules. So again, I'm going to skip this slide. We all know about the drought issue. This is the occurrence of drought events in the long season water stress over the last 13 years. It's a big problem. I'm not going to insist. Let me just move now to another one. So our research strategy has been very much into the hypothesis-driven approach. We are not looking into things without having a very good rationale. I'm just picking now on a very obvious example where the strategy will be about water saving through somehow controlling the guard cell aperture and closure. And as you're certainly familiar, I'm not going to describe the process. For sure, there is an important plant hormone which is involved in this whole process, which is called abscisic acid, as you all know. And we have been interested, given the recent breakthrough from Professor Sean Cutler, on actually understanding and elucidating the target of ABA in the ABA signaling pathway. We have been collaborating with Professor Sean Cutler, and actually I'm going to tell you a bit more about this, because it is really a very exciting piece of work which is currently undergoing and allows us actually to to make some very interesting breakthrough discoveries here, both from the chemistry standpoint as well as from the genetic standpoint. So bottom line, as you all know, plant hormones and certainly ABA are often, of course, involved in many different physiological processes. We know that ABA is involved in seed germination, it is involved in root growth, but it is involved as well in the response of the plant to abiotic stress. So certainly if you do apply ABA, you don't want to have these very different responses of the plant to happen. You want really to be very selective. So what is underpinning this, of course, is a vast number of ABA receptors. And here we're talking about the knowledge on the corn plant, which is the plant of interest and not Arabidopsis. We have been able, together with Professor Shankatla, to actually elucidate the receptors, the isoforms of the ABA receptor, which are involved in the response to drought. In other words, you can well see what's the next step. We actually are able to design selectively molecules which are ABA agonists, which are mimicking the effect of ABA, but that are only acting on these specific receptors. So this is quite exciting, I have to say. You have here a picture just showing a controlled soybean untreated submitted to water stress. And you can see that on the treated, this is our best compound we have currently. When we treat the soybean, sorry, I think you can't see the pointer for some reason. Yeah, when you treat the plant with the compound, you can well see that it copes much better with the drought event. So this is a molecule which is applied in a foliar spray and which is somehow closing the stomata and saving the plant is by this means saving water. In a very simplistic description, obviously, we all know that there are other impacts on photosynthesis depending on how much you do close the stomata, and these are all things that we are carefully looking into. So now, making the story short, with this knowledge, as I said, we can specifically design chemistries which are binding to these specific receptors of interest. We can as well engineer the right, as I call here, corn receptors. And this is an example of one of our events we have in our portfolio. And you can well see that this plant, so genetically modified, is actually using 40% less water as compared to the non-GM modified crop. So this is all early days, but I have to say it's very promising in terms of first stage. So let me move on into give you a broader sense of what else do we have in our research portfolio. As I said, and apologies really to come with those very, let's say, non-specific descriptions of mode of action, but I think you can probably well understand the reasons why. We have a number of projects in our portfolio of chemistry addressing drought, as I mentioned, by manipulation of ABA signaling pathway. This is the one I referred to before. We do have as well other pathways which were selected to address the same problem through different strategies. We have as well stress tolerance. We have a project where we are looking into as well a specific mode of action as well as cold stress. So in essence, we do have a quite focused portfolio. Given the complexity of every single project, we really try to go deep into the science. And I have to say we have been extremely lucky within Syngenta to be able to have brilliant people and scientists with all the knowledge we need in a very diverse field disciplines to be able to get to the point where we are. Going back to my slide here, so this is just to exemplify that we do have a chemical class which have potential for cold stress mitigation during corn seedling establishment. You know that changes in farmer practices and early planting has a sort of a side effect which is unfavorable because seeds get into the soil in a soil which is and exposed to cold, and this has a significant impact on the yield at the end of the day. So making this short, we are able, through our different greenhouse experimental assays, we are able to assess on whether we have chemistry which actually is able to impact this very nice sigmoid curve here that reflects somehow through the slope, the kinetic of germination, through the plateau, the homogeneity of the seedling establishment, as well as the T50, which will be the time that half of the population has emerged. And those are physiological parameters which are important to us in order to characterize whether the chemistry is impacting on this process or not. So turning now to the right of this slide, you do have your chemical class with potential for drought mitigation. As I said, we're looking into as well a strategy which is more about preparing the root architecture, have better roots, which of course the word better has to be translated in physiological traits that we are carefully looking into. But I'll stay for today at this level. But we just to exemplify that we are well equipped and set up with assays which do allow us to measure in a very great detail impact of chemistries on the root architecture at different subtraits, laterals, and soil volume explored, and many other things which are of interest to us. Of course, not to mention the different levels at which the roots are changing from the top of the soil to the lowest layers, which are as well an important trait. So this is really to exemplify what we do have. So in chemistry, now let me move into the genetic part. This is to mention our native trait, Tesian. Maybe some of you are familiar. We do have these lines, these hybrid lines in the market since 2010 in the US market, and now we are expanding this to Europe as well. So interestingly, those the traits and disadvantage we have here is really linked to a number of selected genes which are underpinning this beneficial trait related to drought mitigation. So it provides a season-long drought protection, but interestingly as well, it can deliver up to 50% more yield under drought conditions, and it also maintains a very good yield when there is no stress. So it is really it's been extremely successful in the market in terms of this performance. As I said, we are expanding to Europe, and here I just profit to make a remark. We are using very much in all this area of research the skills we have in our environmental crop modeling team. They allow us really to understand how to position our products. In other words, we do, through the team is doing a lot of data mining and analysis, correlating with weather and trying to really understand what are the places in the world in relation to market regions where the problems really are occurring most frequently, and this is the place where we should actually position our products. And here you can well see that we are guided by this analysis. We're going to position our new hybrids in very specific areas in Europe and east of Europe, CIS as we call. So we as well using different technologies, drones being one, that are equipped with cameras and allow us, for example, if they are equipped with thermal cameras, to be able to sense, what is the temperature of the canopy and what's the level of stress. It also allows us to select our best hybrids through these means and the field, right? Okay, good. I'm sorry, could you please tell me about time? I'm a bit... 5 minutes. 5 minutes more. Okay, so I have to speed up then. Okay, so this is just to give you one example now. As I said, this is again the ABA pathway. We have been, as I said, already looking into GM events where plants can better tolerate limited water compared to control. And you can well see, again, very early days, but quite promising. So I'll skip this because it's very much a repetition of what I was telling you. Importantly, we do have quite a significant number of events which prove so far to be performing in this greenhouse environment that we are pushing to the field to see whether the performance translates in field conditions. So with this, as I said, of course, we need to have the proper expertise and the proper capabilities to be able to move on with these projects. And what I'm going to show you briefly in a very high-level description is what are state-of-the-art phenotypic capabilities. This is what we have in my group has been building since the last, say, 5 to 6 years in Stein, Switzerland. This is just a selection of a number of assays we do have. The first one has to do with the germination robot, which allows us to measure the impact of small molecules or even differences across a panel of genetics on the kinetic of germination. So this has been developed for corn seeds, and we're expanding across crops as well, and it's based on the 24-well plate, and I proved to be extremely useful to somehow test the first as a proxy to test for compounds which will impact on seedling vigor under stress or under normal conditions. So then we do have a number of assays which allow us to progress into a more realistic or agronomical, meaningful environment, of course, which then is to go into an emergence assay. We are able to detect as well chemistries with impact on root architecture, and this is a very familiar hydroponic root system which has been developed for rice as a plant model, that we can then progress to our rhizobox assays, and we have small rhizoboxes, and then we go to big rhizoboxes. This I didn't mention here, but you saw in another slide. We equipped with a LemnaTech as many other companies, and this allows us to both assess effects on shoot as well as on roots from chemistries or other technologies. We are equipped with a very performing, let's say, system based on balance, which allows us to measure transpiration rate. This is all those different assays are done under a controlled environment. Last but not least, we are equipped with a couple of different things. Just to exemplify here, we have an high content screening platform, and this allows us to somehow dissect the phenotypes. We can see, for example, if we do have a molecule which is changing the root architecture, whether this is impacting at the tissue level, the elongation zone, the division zone at the root tip. And so it's a very good way to somehow dissect phenotypes. This is a technology which is actually broadly applicable in different areas of our research, not only abiotic stress. And finally, we do have chemical genetic screens, which allow as well for deep phenotyping and mode of action understanding. So we have a number of those which we are developing in systems like corn, and it has been quite an interesting challenge to have. So we have, as I said, the global platform. I'm just referring the previous slides to what we have in Stein. We do have as well 2 additional important sites in our organization which are equipped with plant screening platforms in Beijing, as well as in Research Triangle Park, with most of the work done on GM, as well as genetic and genome editing is done. And you can well see that we have state-of-the-art greenhouses. We also equipped with a system of balance to assess water use. And interestingly, we have these very unique precision growth chambers, which essentially were designed for NASA in the US. And then I think we are the only ones who are actually bringing this technology to the industry to measure quite with precision things like CO2 and any kind of exchange between the plant and the environment can be monitored in these chambers. So with this, of course, greenhouse is all extremely important to us, but the translation of the performance we see to the field is mostly important. So just me give you an example. We are as well investing capabilities in the field, and this is an example of managed stress environment we do have in order, of course, to assess, as I said, our different technologies. So this is an example in the US, in California. We do have one site as well as in Colorado. And this is just to exemplify that, we do have developed very specific, robust protocols which allow us to achieve the goal, which is, for example, in this case, I think to achieve 40% yield loss and a limited irrigation. And then, of course, you assess within this controlled environment how those hybrids or your chemistry is performing and helping decreasing the yield loss in a sense. And this is, of course, quite unique and really important before we go to a network of field trials in the marketplaces. With this, this is just something I mentioned already. I'm probably going to skip. We do have high precision field phenotyping capabilities. In this example, I'm just referring to, again, it's all about thermal imaging and how we can assess the stress level and how the plants are coping with stress after application of chemistry or through genetic means. So with this, let me just summarize. As I said, we are in the place where we have now state-of-the-art capability for phenotyping in a controlled environment. We do have as well capabilities in the field, so I think we're very well equipped to be able to push forward those different pipelines, both in chemistry and genetics. With this, I'm just going to briefly give you, for those who are not familiar with Syngenta, these are the different sites we have in R&D, or quite spread all over the place. We have some major sites, Gelatil, we have Stein, we have RTP, and we have many other sites which are as well extremely important to us in support. So this is where I'm located and my team. Switzerland have a couple of activities. You know, we have the global headquarters. This is a picture from Stein, where we do have R&D. We have a formulation process just across the road, and some other places where you have production, sales, marketing, and field trials. So with this, I'm going to finish by acknowledging my team. Here we have Raymond, which has been extremely instrumental in all the germination development. She's a plant biochemist. Michael Courbeau is really focusing his effort on root and root-related traits. Michael Bartsch is looking into drought and dealing with all the drought-related aspects and transpiration. Jörg Leipner is and his team is looking into emergence and rice heat stress. So everyone is very much busy both on capability development as well as project support, and as project biologists, leads, they are all extremely engaged in these all activities on the chemical side. So then we do have our agronomical traits team, which is led by Nick Bate. We have our environmental crop mobility team, which, as I said, is been extremely important to us to help positioning the products in the market. Without biometrics, we wouldn't be here. The design of the experiment is really critical to the success and the confidence we have in what's coming out from our assays. And last but not least, our chemical team led by Sebastian van der Boon, as well as the automation team, which is helping us setting up those platforms. So with this, I'm going to stop here. Thank you very much for your attention. I'm happy to answer any questions.